Hi everyone, thank you for coming to my talk today about our recent work in modeling glioblastoma stem cell lines. First, a bit of background. Glioblastoma is a highly fatal form of brain cancer. While there are treatments available, there are no targeted therapies, even though there has been substantial work on classifying these tumors into clinical subtypes. Any attempt at developing a targeted therapy is confounded by the existence of cancer stem cells. These cancer stem cells are able to evade conventional treatment and lead to tumor relapse. Another factor that contributes to treatment resistance or lack thereof is heterogeneity between individual patients. And so, three glioblastoma patients, their tumors might have vastly different genomic profiles. And so, we can see a real world example of this in the figure on the right, where we show cell growth for three different glioblastoma stem cell lines in response to a wide array of chemical kinase inhibitors. And so, you can see that for two cell lines, they are not guaranteed to both respond to the chemical kinase inhibitor in the same manner. And so it's clear from this that our understanding of heterogeneity is incomplete, and we need more work to understand how heterogeneity in glioblastoma stem cell lines influences treatment susceptibility. We propose utilizing computational modeling to study heterogeneity in glioblastoma stem cell lines. Specifically, we'll be using a discrete model, which is able to consider both dynamics and concentrations of model elements, but requires less prior knowledge about kinetic parameters than ODE-based models. Our goal with modeling heterogeneity is to be able to better predict kinase inhibitor susceptibility on a cell-by-cell basis. And so here we present a discrete glioblastoma stem cell line that's informed by individual cell line data and is able to accurately predict kinase inhibition susceptibility in these individuals' glioblastoma stem cell lines. And so the first step in model assembly is to identify nodes that are likely involved in glioblastoma stem cell signaling. And so to identify these nodes, we can go to experiment databases. From the Cancer Genome Atlas, we can find genes that are commonly differentially expressed or mutated. We can also utilize PubMed to find likely genes and proteins from highly supported publications or novel publications. Once we have our list of nodes likely involved in glioblastoma stem cell signaling, we need to identify the edges between those nodes. To do so, we can use pathway databases like Reactome, where we take our list of involved model nodes, and we can find upregulated canonical signaling pathways. Once we have our canonical signaling pathways, we want to identify directed interactions between our model elements. The reason that we want directed edges is because we want to study the mechanistic interplay between different model elements. And so once we've identified direct interactions between our model nodes, we then are able to write update rules. And so this is for a discrete model. So we show a toy discrete model in the upper right-hand side. The nodes are model elements and the edges are interactions between them. We show both negative and positive interactions where the activity of the downstream element is either inhibited or activated. And so we showed two sample update rules below that for two of our model elements. And so I think it's important to note that for these update functions, F2 and F7, these update functions could be a wide variety of things. So this could be a min-max function, this could be a Boolean function, weighted sum. And so this is just to show that there's flexibility in how we represent the interplay between multiple regulators and their effect on a downstream node. Once we have our update rules written, we use interaction sources to find support for our model logic. And so we focus on two interaction sources in particular. We look at the intra-database, which contains machine-read statements from millions of papers, and the PCNet interaction database. And so PCNet is a metadatabase of protein-protein interaction networks. And so we use these two sources to fine tune our model logic. And so there are instances where we are able to update interactions based on our interaction sources. So in cases where we've incorrectly represented an interaction in the wrong direction, where the upstream and downstream nodes are flipped, we could have an error in the way we wrote the update function itself, or possibly it could help us inform the effect of an interaction. And so we're also able from Indra and PCNet to find interactions that should be removed. So this could be a case where we have an interaction represented as a direct mechanistic effect, where in reality it's a correlation that was inferred from data. Or it could be an indirect interaction that is already encoded for in the model by several direct interactions, and so it's redundant. Finally, in addition to updating or removing these interactions, using Indra and PCNet, we are actually able to locate missing interactions that could be potentially very important for our model. And there are a couple different reasons we could miss an interaction. This interaction could have only recently been published. It could be canonically implicated in a different context of interest, such as another cancer cell line. And so we're able to use interaction sources to refine our model logic, remove interactions when necessary, or add back missing information. Finally, after we have our update rules, we're able to parameterize our model and simulate. And so parameterization is where we're actually able to capture some of this cell line heterogeneity. So we show an example of parameterization through initializing element activity values from gene expression data. So in the graph on the left, we see the expression for 3 genes across 3 cell lines. If we analyze expression and we find differential expression between groups, we see that in some cell lines, a gene is higher or lower expressed, significantly so than in another cell line. And so to use this information to set starting activity values, we set the number of activity levels to equal the number of cell lines. And so these activity levels are a discrete representation of the expression or activity of a model within an element within our model. And so, for example, for gene 2, cell line B has significantly higher expression in cell line B than any other cell line, and so we initialize that to the highest value, 2. And we initialize accordingly. In cell line A, we initialize gene 2 to 1. And finally, in cell line C, we initialize gene 2 to 0. Once we've parameterized our model and we have a separate set of parameters for each cell line, we're able to simulate the executable model. And so we use the discrete stochastic heterogeneous simulator, or the DISH simulator, to provide dynamic predictions of model element behavior over time. So here I show the results of the actual model assembly. And so within our model, we contain 169 elements and 295 interactions between them. This figure has been edited for clarity. However, we show some of the more crucial pathways and observables within our model. This model contains 159 genes and proteins, 6 biological processes, and 4 small molecules. You'll also notice that we do keep track of the cellular location, or the organelle in which an element is located. And finally, this model is not comprehensive, so it does not contain every possible pathway or protein that might be involved in glioblastoma stem cell signaling. However, it is as small as possible while still giving us accurate and meaningful results. Here we show some of the key cellular processes in the glioblastoma model. We're mainly interested in proliferation, which is directly regulated by cell cycle progression and apoptosis. We use proliferation as a marker for tumor survival. Here we show the results for network verification using Indra and PCNet as described previously. For each of the interactions within our glioblastoma model, we look for supporting evidence within INDRA. And so we find that for 100% of our interactions from our model, there's at least one supporting source in INDRA. And in fact, if we go by the INDRA belief score as a measure of the amount of evidence, we see that most of our model interactions actually have a very high INDRA belief score. So there's a lot of already existing supporting evidence for our model. If we look at PCNet, we find that 74% of our interactions have also been verified by an additional 2 networks contained in PCNet. For 159 of our model elements, we are able to initialize from RNA-seq data. Out of these 159 elements that we've initialized, 39 of them are different between the cell lines. And so we're able to capture some of that cell line specific heterogeneity. Here we show a simple example of validation by kinase inhibition. In our model, we can artificially knock out a kinase. We can observe the effect propagate downstream. And finally, measure its effect on proliferation, which, as I mentioned previously, is a marker for tumor survival. We can compare this to actual in vitro data. And when we do this for all 3 cell lines, for 11 kinases per cell line, we show that our model is 90% accurate when predicting both the direction and magnitude of change induced by kinase inhibition, and 100% accurate when considering only the direction of change induced by kinase inhibition. And so not only is this model highly predictive, it's cell line specific. In the figure on the right, where we show in silico knockout of PDGFR for our 3 cell lines versus in vitro knockout, we show that our model only predicts tumor inhibition for the MGG8 cell line, which is supported by data. In places where we're not able to accurately predict the magnitude of change, only the direction, we find a couple of very interesting cases where inhibitors are just not quite as effective as expected, which is obviously very relevant when we're talking about treatment susceptibility for individual patients. And so we've informed the model to the best of our understanding. So these are actual cases where we have a real opportunity to explore these experimentally and determine and possibly new information about glioblastoma stem cell signaling. And so in conclusion, we present a stochastic dynamic glioblastoma stem cell model that includes key pathways and proteins, is highly supported both by database and literature resources, is initialized on cell line specific data, highly predictive. In 90% of cases can predict both direction and magnitude of change induced by kinase inhibition, and is able to explain treatment susceptibility between cell lines. With that, I'd like to thank everyone who worked on this project in both the Melody Lab and the Cochrane Lab. And with that, I'll take any questions.